Assalamu alaikum all. Today is the last part in sugars or carbohydrates, which deals with polysaccharides. So we discussed earlier the structure of monosaccharides and their formation of larger structures like disaccharides and oligosaccharides. Whenever we have a very large number of monosaccharides connected together, they're going to form polysaccharides. These polysaccharides might be of the same monomer residue or same monomer type. For example, if it's all made of glucose, we consider it as homopolysaccharide, whereas if it contains more than one type of monosaccharides or monomers, we consider it as a heteropolysaccharides. These polysaccharides differ in their monosaccharide unit that is repeated inside them. They're different in size, they're different in length, they're different in branching, how often does it happen, and how many layers of branching we may have in a polysaccharide. They have different functions, they have different sources. Some of them are just present in plant cells, others are present in our cells, others we can deal with them in terms of digestion, we can deal with in terms of digestion, others we cannot, etc. We'll discuss inshallah in a minute some examples on these polysaccharides, their structures, functions and features as well. So let's start first with glycogen, or the storage form of sugar. So whenever we have excess sugar in our bodies or in our cells, they're not going to be stored as glucose or sugars, they're going to be stored as glycogen. Glycogen is a polysaccharide, very complicated in structure. It is made of glucose unit as a monomer, as you can see in here. This is the glucose, as you can see in the picture. This type of polysaccharide is a branched one, as you can see in here, and it has so many layers of branching that may reach 13 layers, so it may branch 13 times. And this branching happens very often. It's like every 10 residues, every 10 glucose residues, there would be a branching point. If you notice, this is the first glucose, this is the one glucose, and this is the other glucose. This is carbon number 4 from this residue, and this is carbon number 1, so it's an alpha 1 for linkage, and the V-shaped V-shape indicates this type of linkage. And notice that it is going to be repeated along the main chain as well as the branched chain. The only difference in terms of type of bonding occurs on the branching point itself, because we already have the residues in the main chain making these bonds between carbon number 1 and carbon number 4, so we have to look for another option to make this branching happen, and the best in this situation is to invest carbon number 6, since it is out of the ring, so this would avoid us the steric hindrance that may happen. So carbon number 6 of the main chain is going to interact with carbon number 1 in the first residue of the branch, making an alpha 1,6 linkage between them. Then afterwards we're going to end up, or we're going to continue in alpha 1,4 linkages until we reach the end of the branch. Notice that for the main chain and for the branching as well, that the for example, this residue, the last one, if we consider this residue the last one in the chain or in the branch, and this one the last one in the main chain, notice that the free carbon in this residue is going to be carbon number 4. Carbon number 4, because carbon number 1 is taken for the bond with the previous residue. Same thing in the main chain. So all the ends of glycogen actually end with carbon number 4. So that's why we call them, these red balls, we call them as non-reducing ends. If you look at this picture, so they're all the red balls at the ends of the branches and the main chain are considered as non-reducing ends, because the anomeric carbon, or carbon number 1, that is important for oxidation reactions or for a sugar to become reducing, is already occupied in the bond, so we would only have a free one just at the very beginning of the main chain, and this one is definitely not enough to make glycogen as a whole, as a very complicated and large molecule, reducing. So it is a non-reducing sugar. These glycogen molecules are going to accumulate in glycogen granules in the cytosol of many cells, actually, and all cells of the body, but they have very large stores, or the largest stores, in the liver and muscle. Since these organs or tissues are very vital and need a larger amount of energy compared to other tissues, that's why they have the largest stores or glycogen stores and granules in them or inside them. The next polysaccharide is starch. starch أو النشا يعني is a plant polysaccharide, so the source of it is plant cells, but we don't synthesize them in our cells, but we can obtain them from diet. And there are different sources of starch, like rice, and corn as well, as a source of starch. It is composed of two different forms of molecules. The major one is the amylopectin, which constitutes around or more than 80% of the starch composition, whereas the minor one or minor component is the amylose, that constitutes around 10 to 20%. The amylose is made of long chains of again glucose monomers. Again, they are connected via alpha 1,4 linkage repeated along the chain. They form helices similar to alpha helix, but alpha helix, you remember, are present in protein structure, but I mean similar to the shape of that helices. And they don't have the amylose does not have branching in their structure, whereas the amylopectin, as you can see in here, has long chains as well as branching, although they're not as common as in the glycogen that branches like every 10 residues. In this one or in the amylopectin, branching happens less frequently. It happens like every 25 residues. Again, for the idea of reducing and non-reducing, the free ends at the ends of these red balls at the ends of the branches and main chain, they all have the carbon number 4 free. That's why they're considered as non-reducing ends, except for the very first one in the main chain that but this one is not going to affect the very large and complicated structure of starch, and its effect is negligible. That's why the whole sugar or the whole polysaccharide is considered as non-reducing. If we break up these units, I mean, like if we cut two residue unit, this results in the formation of the maltose disaccharide in its structure, and this is what actually happens during digestion of starch along the GI tract, producing these disaccharides, and at the end they would be broken down into their forming or their constituting glucose residues for absorption to happen. So the starch comes from a plant source. Glycogen is synthesized in our cells, okay, and is present in animals. That's why it's considered as, or it's named as, animal starch. So let's compare between glycogen and amylopectin, which is the branched form of carbohydrates or polysaccharides, of starch. So they're both made of glucose, as we've seen earlier. They both have alpha 1,4 linkages in the main chains and alpha 1,6 at the branching points. For the glycogen, as I said, is existent in the animal cell, whereas amylopectin is plants, but we can digest it once we obtain it through diet. Glycogen, in terms of branching, is much highly branched than the amylopectin. As I said before, branching happens every 10 residues, whereas it happens around every 25 residues in amylopectin. So what is the importance of branching? Why would we have branching, as we've seen earlier? And for starch and even for glycogen, and why we would have more branching for glycogen. So as I said before, for glycogen, it is the main form, or it is the form in which excess sugar or excess glucose is stored in our cells. It's not going to get stored as glucose. So whenever we have this complicated and highly branched structure of glycogen, this would increase the efficiency of breaking down this molecule once we need these glucose residues as a source of energy. So there would be an enzyme molecule on this end, another one on this end, another one on this end, another one on this end. So there would be so many ends for the enzyme molecules that break down and separate these glucose residues from glycogen structure. Okay, so once we break them, we're going to release a large number of glucose residues or monomers in a time unit. Okay, and they can be combusted, and they can enter into the glycolytic pathway, producing the needed energy. If we just have few branches, there would be few ends and less efficient production or release of glucose for energy production. It also, or the branching also, increases the solubility of these molecules. Another type of polysaccharides is Dextran, which is considered as a storage polysaccharide, and this one specifically is important for you as dental students, because this type of polysaccharide is synthesized by bacteria and yeast, and it would create a complex network of molecules that can be laid down on the tooth surface. Okay, so bacteria can colonize these the networks of molecules and stay there for a longer time, and acting on teeth to facilitate, destroy, and damage, and forming caries or decay in teeth. Okay, it's made of glucose again, but this time these glucose residues are connected through the carbon number through alpha-1,6 linkages, you can see in here, and they have also so many branches, and they can be of different types: 1 to 2, 1 to 3, 1 to 4, etc. So this creates a very complicated and highly branched polysaccharide that acts, as I said before, like a network or like a meshwork in which a bacteria can grow easily and colonize to facilitate its release of acids and products that results in tooth decay. Another type of polysaccharides is cellulose. Cellulose is polysaccharide that is made of again glucose connected via beta-1,4 linkage, because the monomers are actually beta glucose. They are made of straight chains. They don't have branches at all, and they are lined along each other, so they would have a very complicated structure that forms that's made of cellulose chains, as you can see here. This is one chain, another parallel chain, and whenever they are very parallel and highly compacted, as you can see here, there would be more non-covalent interactions between them, specifically the hydrogen bond, and this would make their structure stronger or better in terms of mechanical properties. These would assemble into microfibrils, and these microfibrils assemble into larger structures of macrofibrils, and then they make the cellulose fibers that make up the structure of a tree and structure of plants. Sorry. So the structure of plants, for example, ساق الشجرة يعني مثلا بتشوفوا بيطلع منه هيك زي الخيطان لو اجيت تقشر منه شوي، أو أي حتى نباتات صغيرة مش شرط تكون أشجار. That's why so the whole supporting structure of a tree or of plants is made of these cellulose fibers. So we need them to be mechanically strong and with high mechanical properties to be able to perform its structural role. Okay, so the disaccharide subunit that makes the structure of cellulose, or I mean whenever we have two monomers or two glucose residues connected together via beta-1,4 linkage, this molecule is called cellobiose. This cellulose cannot be digested by our GI system because we don't have the enzyme cellulase in our intestines to break down this beta-1,4 linkage in the between glucose residues. That's why whenever we ingest cellulose or cellulose fibers يعني الألياف بالأكل, and specifically in fruits and vegetables, they're going to stay as they are, not digested in the intestinal tract. So what happens next is that they would attract water molecules towards the intestine, okay, to maintain the osmotic pressure and get it back to normal levels. This would result in facilitating the getting rid of the waste products, okay, or of feces. عشان هيك, that's why the people with constipation are advised to eat more fibers. Okay. Another thing or another importance for cellulose, that it acts like a meshwork or like a network that can fish the cholesterol molecule, toxins, etc. So they do not get absorbed by the intestinal over the intestines, and we get rid of them. We get rid of them into the feces. They also, because of their high content of glucose, that is highly polar molecule, they're going to attract water molecules interacting with them through hydrogen bonding. This would enlarge the structure, so they would fill up the intestines and give you a feeling of that you're full and you don't need to eat. That's why it's again among the advices to people who want to lose weight. Okay, so cellulose is highly advantageous and beneficial for our bodies because it does not get digested. But the enzyme cellulose is present in the intestines of some animals like cows and sheep, etc., who rely mainly on plant sources as their food or nutrition, and they can digest it. But in our intestinal system, we don't have this enzyme. Another example of polysaccharides is the chitin. This one again is a structural polysaccharide. It's present in the exoskeleton of different animals, okay, and it's made of repeated unit of N-acetylglucosamine. So this is an amino sugar, as you remember, with this modification here, an amino group and acetyl group, and this one is repeated, and these monomers are connected via beta-1,4 linkage again. Another example on polysaccharides is pectin. Pectin is present in or produced by plant cells and by bacterial cells, and it's made of this modified sugar, as you can see in here. So what are the modifications, and what is the sugar first? It is galactose, as you can see in here, because OH group on carbon number 4 is directed upwards. Okay, the linkage is of a beta type, beta-1,4 linkage, as you can see in here, and it's made of two sugars, this one right here and this one right here. So this is a modified sugar, an oxidized sugar, and this is another one, an esterified after oxidation. So it's a heteropolysaccharide, it's not a homo. So far all the examples we've discussed are homopolysaccharides because they're made of single type or one type of monomers. This one is made of two types of monomers, but they're derivatives of galactose, as you can see in here, and they're connected via beta 1,4 linkage. Pectin is important or is used as a gelling agent. So you've heard about the الجلو النباتي صح؟ This type of jello is we use in this type of jello pectin as a gelling agent to make it become a gel-like material. يعني العامل اللي بيخليها تصير هلامية أو تتحول للحالة الهلامية هو البكتين. And whereas the so-called هو الجلو الحيواني, we use gelatin as a gelling agent. Okay, gelatin is a different molecule, different in structure. Its source is animal. It comes just from cows and pork, and pigs. And it has different properties in terms of the setting. How does it make the jello becomes in this gel-like or becomes gel-like material. هيك يعني يصير مادة هلامية. بتختلف تعرفوا. One of them you need to put it in the fridge, another one it can just set and become jello or gel-like material just at room temperature. So we've already talked about the some chemical properties of monosaccharides, disaccharides, and some of the polysaccharides, and I've discussed with you that the free ends of, for example, glycogen and the starch or amylopectin, they all have carbon number 4 as free carbon. So that means they're not going to be reducing sugars because these are not anomeric carbons. They cannot get oxidized. The anomeric carbons are already occupied and they are bonded through the different types of bonds in different types of polysaccharides. So generally polysaccharides are non-reducing and they would test negative in the oxidation tests that we discussed before. Another type of sugars or polysaccharides is the glycosaminoglycans or GAGs. The glycosaminoglycans are different types of polysaccharides made of 2-unit or disaccharide units repeated so many times in this polysaccharide structure. So they are heteropolysaccharides, they're not homo. The sugars that made up the repeating unit or in these glycosaminoglycans has or the repeating units has a negatively charged carboxylate, so they're either oxidized sugars with carboxyl group that donates the proton and becomes negatively charged, or they may have sulfate groups, as you can see in here. So they may have sulfate groups, as you can see here, or carboxyl group. And they also have so many of the amino sugars, galactosamine, glucosamine, etc. So they have to have highly polar monomers in their repeating units, either by the sulfate group, it gives a negative charge, carboxyl group, a negative charge, also the glucosamine or the amino group that is highly electronegative and makes the bonds around it polar, etc. So they are highly polar. So that's why they're going to attract water molecules towards them, specifically that they are present in the extracellular matrix. So they're present in the ECM or extracellular matrix of different tissues. They attract water molecules towards them, okay? And they would become larger in size and acting like cushions زي المخدات يعني in shock absorption. يعني واقي صدمات أو بتحملوا الصدمات. So this would make your cartilage, for example, not getting broken easily whenever it gets hit or traumatized for whatever reason. Other different tissues also have different types of glycosaminoglycans. for performing different functions. Let's have some examples. You're not required to know all the details like the composing monomers and their names, but you have to know their general properties. So again, polysaccharides with disaccharide repeating units. These disaccharides are made of monomers, and these monomers are modified sugars, either with carboxyl group, sulfate group, in addition to the amino group. Okay. So, for example, chondroitin sulfate glycosaminoglycan is made of glucuronate and N-acetyl galactosamine 4-sulfate. Dermatan sulfate is made of iduronate and N-acetyl galactosamine 4-sulfate. Hyaluronate is made of glucuronate and N-acetyl glucosamine. Chondroitin 6-sulfate, same thing as chondroitin 4-sulfate, but the position of the sulfate group is at carbon number 6 rather than carbon number 5. Heparin is also made of glucuronate 2-sulfate and sulfoglucosamine sulfate. The Keratin sulfate is made of galactose and N-acetyl glucosamine 6-sulfate. So you can tell that these monomer units are derivatives of glucose and galactose, in addition to this iduronate that is present in dermatan sulfate. So these are again the examples that we discussed earlier with their localization and different functions. For example, the hyaluronate is present in the synovial fluid, or the fluid that surrounds joints to lubricate these joints. In addition, it's present in the vitreous humor in the eye, okay, as well as the extracellular matrix of loose connective tissue. Its main function is to lubricate lubricate the joints, for example, if it's present in synovial fluid, and this would make them more shock absorbing. بيتحملوا الصدمات أكتر. Okay. And you've probably heard about the hyaluronic acid or hyaluronate that is present also by that is used by dermatologists and some dentists actually to be injected for cosmetic reasons. Another one is the chondroitin sulfate that is present mainly in cartilage and present also in bone and heart valves, and chondroitin means that it's present in cartilage, and it's actually the most abundant gag generally, and it makes, as I said before, these different tissues bear load and bear some trauma without getting broken easily. So cartilage, bone, and heart valves, they get heart valves, for example, get exposed to pressure of that pushes the blood into the different parts of the body. That's why it needs to stay as it is, or it may get some distortion, but it gets back to its original position or original shape after this pressure is removed from being on it. Sorry. Heparin sulfate is present in the basement membranes underneath the epithelial cells, as you'll see or discuss in histology course. It also present in the as a component of cell surfaces. Heparin, as you know earlier, I'm sure, it acts as a natural anticoagulant and present in different types of cells like mast cells. Dermatan sulfate is present in dermatan, so this means that it's present in skin. It's also present in blood vessels as well as heart valves. Keratin sulfate is present in cornea, bone, cartilage, etc. And it again makes these tissues more resisting to trauma, or to getting broken by different types of trauma, or distorted by different types of trauma or pressure or forces to which they are exposed. This one specifically, or the keratin sulfate, aggregates with chondroitin sulfate. Okay, with chondroitin sulfate, and you can tell that the localization or the sites in which chondroitin sulfate is present, we also find keratin sulfate. These glycosaminoglycans as polysaccharides are going to associate with a small protein part. It's just a small, so making a structure or the structure of proteoglycan, and as the name implies, it's a proteoglycan, so the protein is the minor component and the sugar is the major component, just like the family name. So you have the family name at the end, and it's actually the major component, the polysaccharide, and this is the individual name or the first name with the minor component or the smaller component, which is the protein. These different types of proteoglycans, again, they are different in the extracellular matrix as a structural component of different examples of connective tissue. Their main action, again, is to lubricate through their GAG component or sugar component. They also are essential for adhesion and interaction between cells, and they also can bind and interact with the outer environment surrounding cells and stimulate cellular proliferation. So again, this is the, for example, the chondroitin sulfate, okay, and this is keratin sulfate that associates with it, okay. This one here, the red one, the red long line, represents the protein component, making this structure of proteoglycan. So this is again a zoom in. Chondroitin sulfate, keratin sulfate, core protein, and they are connected to a backbone like hyaluronic acid, making a very complicated structure, as you can see in here. These proteoglycans change a lot with the differences in the outer environment or the surrounding environment, again, around cells. For example, these branches, just like brush bristles, زي فرشاية الرضاعة البيبي هاي اللي بنطفوا فيها الرضاعة البيبي إذا بتنتبهوا. We can change the amount of branching. So if whenever we have less bristles or less branches, so their density is lower, so there would be more spaces in the extracellular matrix for transport and passage of other molecules. But if we have the more dense under certain conditions, we have like we're blocking the way against the movement and easy transport of different molecules in the ECM. So depending on the situation and condition to which the cell is exposed, there will be changes in the extracellular matrix and changes actually in the structure and complexity of proteoglycans. Another example where polysaccharides are important as structural components is in the bacterial cell wall. Okay, and in the bacterial cell wall there is a polysaccharide, a heteropolysaccharide actually, that's made of N-acetylmuramic acid and N-acetylglucosamine. Okay, so this is the N-acetylmuramic acid, as you can see. Again, it has the component present in acetylglucosamine, in addition to this extra unit on the OH group. So this is the OH group on carbon number 4. We lost the hydrogen and replaced it with this 3 carbon unit, and it has a carboxyl group. That's why we call it N-acetylmuramic acid. The other one is N-acetylglucosamine, NAG and NAM. So this is the lactic acid added to the structure of N-acetylglucosamine to make it into N-acetylmuramic acid. So let's see how these are organized. You can tell, or you can look at this picture. So this is N-acetylglucosamine and acetylmuramic acid. So they're alternating, one of this and one of this, one of this, one of the other one. Okay, and they are connected by oligopolypeptide for cross-linking. Let's have another picture that is actually more clear for the structure of bacterial cell wall. Okay, so these are again the polysaccharide made of the two types, N-acetylglucosamine and acetylmuramic acid, N-acetylglucosamine and acetylmuramic acid, and they form these long chains that sit parallel to each other to strengthen this structure. Okay, so these are all sugars and they're polysaccharides, but they get cross-linked through using peptides or using short chains of amino acids. The whole structure is considered or is named as peptidoglycan. It's not a proteoglycan because these are just peptides. They're not long or large proteins, and they're considered as peptidoglycans. The main component is sugar, but the minor component is just peptides, short chains of amino acids. So let's look at the structure of these peptides. So first, look at this one. This one is the muramic acid. From muramic acid, extending 4 amino acids: alanine, glutamine, lysine, alanine. Okay, so these are 4, to which are attached to number 3, which is lysine, 5 glycine amino acids or residues. 5 glycines from number 3, 1, 2, 3, 4, 5. And the last one interacts with carbon, or the last glycine interacts with the last amino acid, which is alanine, in the adjacent chain. Okay, then number 3 again interacts or is connected to glycine number 1, then 1, 2, 3, 4, 5. Number 5 is connected to the last one, or number 4 of alanine, connected to muramic acid. So this is going to happen across all these chains, connecting them together or cross-linking them together, making this structure stronger. That's why you remember when you discussed in Biology 101 the structure of bacterial cell wall, and the cell wall is actually, or some details about it, sorry. And the structure of this bacterial cell wall is highly rigid, and it's actually not present in our cells as animal cells, but it's only present in the bacterial cell walls as well as plant cell walls, because of the structure of, or complicated structure of polysaccharides cross-linked through peptides, making the complicated structure of peptidoglycans. Let's talk about glycoproteins as molecules that contain sugar component. Okay, they're not polysaccharides, and they're mostly actually oligosaccharides connected to protein component. This time we're saying, or we're naming them as glycoproteins, because the main component is actually protein, and the largest component is actually protein, and there is a minor component or a smaller component made of sugars or carbohydrates. These sugars can be added to proteins, specifically cell membrane proteins, through different ways, either through nitrogen atom, okay, as you can see in here. So these are amino acids, part of the protein structure, and this is asparagine amino acid that you'll discuss its structure later on. And through its nitrogen that is present in its R chain, it's going to get connected to the sugar component, forming N-linked glycosylated protein. Whereas in another type of glycosylation may happen, or adding the sugar, through serine or threonine amino acid, or even hydroxylysine. So the OH groups in these amino acids can be connected to the sugar components to add these sugars to the proteins through O-linked glycosylation. So we have two types depending on the connection point, whether it happened through an oxygen atom or a nitrogen atom. What about the significance of these and the importance of these glycoproteins? The addition of the sugar component to these proteins can be either for soluble proteins, I mean proteins that are present in the extracellular matrix as free proteins, or as or secreted proteins, or the proteins that are present in the cytosol or different organelles not connected or attached to membranes. They can also be found or attached to membrane proteins, and this would support the function of membrane proteins. The addition of these sugar component may have different roles or functions or purposes. The first one is that their importance in protein folding. The presence of the sugar component may guide or facilitate the folding and formation of the final 3D shape of a protein. And if you remove the sugar component, the protein won't fold to its native and functional 3D shape. Another function is or purpose is for protein targeting. So, for example, if you're going to if the protein is synthesized inside the cell, as you discussed probably in the cell biology course, and this protein is targeted or should go to the cell membrane or should go to the mitochondria or go to the lysosome. So many of the localization sequences or signals are actually made of sugars and carbohydrates coming out of glycoproteins. They also may affect the half-life of proteins and prolonging the half-life and functionality of the proteins. They can also facilitate cellular cell-cell communication because the interaction between different cells may happen through these sugar components on their surfaces that are connected to or part of glycoproteins. They are also important for cell signaling. Cell signaling means the interaction between cells and transporting the message from outside the cell to the inside of the cell. And many of the actually receptors that are present on cell surface are glycoproteins, as well as some soluble proteins along the signaling pathways. One last application on the importance of sugars, specifically in glycoproteins and glycolipids, is for blood grouping or the ABO system. There are different ways to group or to type for blood types or blood groups, but this system depends mainly on the sugar, which is the ABO system. For an individual with O blood group, whether it's a positive or negative, that's another way for grouping the positive and negative, or the Rh factor. So the O blood group, on the surface of the red blood cells we have, or the cell membrane of red blood cells, we have glycoproteins and glycolipids that you'll discuss their structure in the lipid we'll discuss their structure in the lipid lectures, inshallah. So this is the structure of lipid, and as the name implicates, it contains a sugar component, and this is actually an oligosaccharide. This oligosaccharide is made of glucose, galactose, N-acetyl, galactosamine, galactose, and then fucose. Fucose is a reduced galactose. Okay, so these 5 sugars are present in a person with O blood group. Same set of 5 sugars or oligosaccharide is present in an individual with A blood group or B blood group, but an A patient or A person has an extra N-acetyl galactosamine, whereas the B person has an extra galactose in addition to the 5 sugars or residues. That's why the person with O antigen or with O blood group can give her or his blood or donate their blood to any blood group, whether it's O, A, B, or AB even, because AB is going to have both of these extra sugars. Why they can donate their blood to any blood group? Because these sugars are present in every one of them. That's why they cannot be recognized as foreign bodies, and they can be accepted by the recipient patient. Okay, whereas if a person with A blood group can donate his blood only to A and AB, because this antigen is present in his blood and the patient with AB blood group. Okay, so it cannot be recognized as a foreign body. For a B blood group, it can be again donated to B and AB, because they have the same antigens and they cannot be recognized as foreign bodies. So this is a minor difference in just one sugar, makes different types of blood groups, and would affect the process of blood donation or blood transfer. With this, we're done with the examples and applications on carbohydrates and their structure as well, and inshallah we'll move on next time for the next topic on the syllabus, which is lipids. Assalamu alaikum.